Ipsita Banerjee. She is really passionate about her science, and I think that it's a good example of things that we hear before from Amanda and Nicholas. So where they develop technologies to see or get these cells, she's coming from a background in bioengineering, and she was working since then in regenerative medicine. Her lab has projects in diabetes and another kind of diseases. So she's taking how the physics, the chemistry, and also the biology can pull all together to create new solutions for diseases that we have. So please give a big applause for Ipsita.
Thank you very much for the kind introduction, and thank you very much for including me in this summit. This has been like a phenomenal meeting. This has been so informative. I'm really excited with this meeting. So as was mentioned, I am in the engineering department. All my training is in chemical engineering and bioengineering. So what you would see is kind of a different perspective of things, and I hope to excite you with the possibilities of engineering inclusion in this regenerative medicine. So what I'm going to talk about is really biomimetic models and engineered organoids with a little bit more emphasis on the engineering. So I wanted to start off with this 2016 publication in Nature Outlook, which kind of illustrated regenerative medicine and demonstrated its potential in rebuilding the body. So I've kept my introduction really very brief because I knew you would get a phenomenal discussion of regenerative medicine and embryonic stem cells, so I can quickly go into the details of my work. So the basis, as we saw in the last two presentations, the basis of regenerative medicine is these embryonic stem cells and the pluripotent stem cells. The promise of pluripotent stem cells being that the induced pluripotent stem cells being that it can be individualized. These cells can be derived from individual patients and from the population, and can grow indefinitely, give rise to most of the organs in the body. So the specific, we work with a couple of different organs, pancreatic islets, and more recently on the thymus. And what I wanted to share with you is our work on engineering the immune system using the thymus. And this is, since I'm an engineer, and all my projects are largely collaborative, so I heavily rely on my wonderful collaborators to support our work. So the thymus project is in collaboration with immunology group in Allegheny General Hospital, Yong Fan and Massimo Trucco. So the thymus is it's a very central and key part of the immune system. What is commonly introduced as the way I understand it as an engineer and an educator is the thymus is a school of T cells. Now, T cells, of course, we are all familiar with T cells and its importance in the immune system, and that it is very well-trained cells that can track down which is a foreign cell and which is myself. Now, where this training happens is in the thymus. What happens is these cells, hematopoietic stem cells from bone marrow, they would travel to the thymus. Within the thymus, these cells called the thymic epithelial cells, and these thymic epithelial cells will interact with these naive cells, which has entered into the thymus, which is not trained yet, and in a cascade of very complex signaling pathways, these cells will get trained in positive and negative selection, and then will exit the thymus and will be trained to do the job. Now, our objective is to engineer the thymus here So what we want to do is, from the induced pluripotent stem cells derived from individual patients, we want to build a thymus so that we can control how the T cell training is undergoing. So that's the main goal here. The way we go about it, kind of the overall workflow, is we will take cells from individual patients, the pluripotent stem cells. These cells will be scaled up in large quantities. We will derive the thymic organoids from these cells. Now, once derived, this can go back to the same individual from which the pluripotent stem cell was derived, or in parallel, we can use this in vitro as a biomimetic twin, either to test drugs on these or even in vitro T cell generation. So that's the overall workflow. So I'll take a step back here and talk about how do we culture the pluripotent stem cells, because the scalable culture is one of the things that I'm passionate about. Now, the most common and most established way of culturing these pluripotent cells is in the tissue culture plastic on the surface of the plastic, which works really well, and it's an easy way of culturing. And what is good about it is it's a very easy way of tracking how the differentiation is going. You can visualize how the cells are changing when we are doing the differentiation. The trouble with this kind of adherent or two-dimensional culture is we are restricted to the scale-up because we are relying on the surface, and it's much simpler if we can make use of the volume of the culture if we want to grow things in large quantities. So that's one problem. The other problem is, since the tissues that we want to derive from these pluripotent cells are all three-dimensional, the differentiation efficiency sometimes is much more reduced when we are looking into adherent culture. Now, having said that, there are ways to culture the cells, these pluripotent cells, in 3D, which is these, as shown in here, which is the stirred suspension culture. And it works, only it's a lot more finicky than the adherent culture. So what happens when we are doing like weeks and months of differentiation, these aggregates that are formed in stirred suspension, they can fall apart or they can coalesce, and we lose the efficiency of differentiation. So one of the things that my lab has come up with is really a very simple way of doing an efficient three-dimensional culture of these pluripotent cells, which can be scaled up quite easily. So what we do is, instead of culturing the cells as is, we embed the cells in these capsules, in these alginate capsules. So what you see here, these are like translucent. What you see here is these capsules are the beads, and the dots here are the single cells that are encapsulated within the beads. Once that is encapsulated, it comes together, forms these aggregates, and once these aggregates are formed, these are pluripotent aggregates, they are stable in culture, and now we can differentiate it to pretty much whatever lineage we want to, or we can. So I've used this culture system for deriving pancreatic beta cells in this configuration. What I wanted to share with you is our more recent work on deriving the thymic epithelial cells in this configuration. So once, again, the entire differentiation happens under encapsulation within the alginate capsules. And what we observe is these cells, it grows to a certain extent, and then it is stable in the culture, and when it is differentiated. At the end of differentiation, now we can dissolve the beads and extract the aggregates. It forms really very stable aggregates. And then we can use it for any of the downstream applications. Or we can further characterize it, as it's shown here, to judge the phenotype, characterize the phenotype of the differentiated cells. What we are doing here is checking its phenotype for the thymic epithelial progenitors. So at this stage, we took a deep dive into the phenotype, and we did the single-cell sequencing of the cells. at the end of the differentiation. And what was really interesting is what we observed was when we are encapsulating and differentiating, the thymic epithelial cell progenitor population that we observe, that we find, has the diversity that is observed in the human thymic epithelial cells, meaning that the thymic epithelial cell is not a single cell type. There are several different subpopulations, and these encapsulated differentiation can give rise to the diverse subpopulation that does not typically happen with the adherent culture. So this was very exciting, and this is kind of an established protocol in the lab now. So having these cells and the organoids, now the next step was, okay, how do we go from the cells and the organoids to the actual thymus? How do we reconstruct the actual organ and see what its function is in the T cell development? Now, to achieve that, what we used was a mouse thymus. So what we need is a scaffold for that. So we took a mouse thymus, we decellularized the mouse thymus to get the architecture of the organ and the scaffold, and then we repopulated the mouse thymus scaffold with the iPS-derived thymic epithelial cells, along with the progenitor T cells. So the idea here is the iPS-derived thymic epithelial cells in this organ structure will now interact with the pro-T cells, the progenitor T cells, and will train the T cells. So this is the scaffold being repopulated with the thymic epithelial cells and progenitor T cells. And what we see is it takes up we pretty much inject the cells and the organoids into the scaffold, and it occupies the entire scaffold nicely. So then to the question of whether it is training the T cells. To address that, what we did was we took this organoid and we implanted it in a humanized mice model. The idea being that can it reconfigure the immune system in the mouse model. And what we observed after a few weeks of implantation in the mouse model could reproduce the human T cell function. So we see good subsets of T cells in the animal model and good diversity of the T cells. So this shows that this concept is working, and we can generate the thymus, and which can train T cells adequately. So moving forward then, what we have demonstrated is it is possible to derive these thymic organoids from iPSCs. But my passion, again, is, okay, how do we get this into a scalable form so that it can be taken to the clinic? How do we scale up this process? So moving forward, then, what we were doing with our encapsulation was really, in the bead formation, was really manual encapsulation. So it's restricted to how much we can grow the cells or how much we can encapsulate. So what we next did was really a simple procedure, but bioprinting these organoids instead of manual encapsulation. So what we are doing is a droplet printing here. It's very simple, and the beauty of this process is in its simplicity, because we can scale up the system we can generate 45 beads per second. And it's not only the speed of the system, we can control the system by this nice air jetting technology, where we can control the size of the bead, we can control the shape of the bead, and we can generate monodispersed spherical beads, which means we can reduce the heterogeneity in the process of bead generation. So again, what you see here is these bioprinted droplets with the single cells of iPSCs embedded inside the droplet. Now, the procedure is, once we encapsulate these cells, these are encapsulated in pluripotent stage. They will grow within the capsule, form these nice aggregates, still pluripotent. And what you see here is that it retains pluripotency, OCT4 and NANOX staining here. And once we have this stable aggregates of pluripotent cells, then we start the process of differentiation. The cells would grow a little bit, and then the differentiation ensues. At the end of the differentiation, we can again dissolve the beads and harvest all the aggregates, use it for downstream application, and do the necessary characterization. So what we have seen so far is the printing process that gives rise to the differentiated cells, just as the manual encapsulation does. And currently for functional testing, we have implanted these in the animal models, and we are still waiting for the results. Hopefully in a month or so, we will see the functionality in training the T cells. So now, that's one part. Now, the other thing is when we look into these cells, as we saw, we need to, when we want to know the phenotype that we observed, that we determined from the differentiation, we will need to get into invasive methods. So we have to fix the cells, stain the cells, image the cells. So it's invasive, and it's difficult to do this online with the manufacturing process. And I'm very motivated from the manufacturing endpoint. So what we wanted to see was if it is possible to develop some quick and simple ways of determining the phenotype of the cells, which will bypass these details of staining and immunostaining. We have to do the imaging, and we can do it with the live cell population. So what we have developed is this deep learning model, where the goal was that I will present the model with bright field images, and if it can classify the phenotype of the cells. So what we have developed so far is deep learning models to identify the thymic epithelial cells and the beta cells. And I'm very encouraged with the efficiency of this process. So what we do in developing the model is we still fix and stain the cells while training the deep learning model. We present the model with like thousands and thousands of these stained images, and then we present it with bright field images that the model has not seen before, ask it to characterize it, and check the phenotype of the cells. So overall, this has really worked very well so far with the beta cells and with the thymic epithelial cells. So we are super encouraged with it. And now, in the next step, what we are doing is we are taking all of this together, the printing and the imaging, and moving forward with automating the whole process of organoid generation and this the manufacturing pipeline. So my graduate student, she is doing an internship now with this company, Advanced Solution Life Sciences, and working with them to develop this automated process where we supply the cells and then kind of press the button, and it will be printed, it will be cultured, media change will be automated, the entire differentiation will happen, and the end, even the imaging and quality control will happen in a fully automated fashion. So really excited about the possibilities here. Now, with this, I just want to leave you with kind of where we are, which is we have a good way of generating the organoids. We can manufacture the organoids. Hopefully within the next 6 months or so, we should be able to automate the process of organoid generation. And then we are really excited about the possibilities. It's just fascinating. As I said, I come from an engineering background, and to be contributing to this field itself is fascinating for me. So some of the techniques, so this thymus, the more I learn, the more I'm kind of surprised by how impactful this organ is throughout different stages of life in different populations. So one of the things was interesting was a 1982 New York Times article which cited the thymus as a mysterious gland which may hold key to aging. So it can have a lot of impact on longevity. What we are focusing right now with clinical collaborators is more like infants and babies and DiGeorge syndrome in the babies, as well as using thymus to promote transplantation tolerance. So that's something that we are currently investing in, but again, we are very hopeful of the possibilities of applying this technology. So with that, I would really like to thank you for your attention, and it was really a pleasure to be in this summit. Thank you.